Hi everybody, it's Dr. Hinkey with the lab video presentation for Lab 13. This is just a quick review of DNA biology. So the structure of DNA and the function, how it replicates during the S phase of the cell cycle. So how we get from a single chromosome to a duplicated sister chromatid, duplicated chromosome to sister chromatids in the S phase, and then how we actually express those genes into a protein. So study guide. Describe the overall structure, the monomers, types of bonds and shapes of DNA and RNA, our nucleic acids. State the components of a nucleotide. That's our monomer. So the polymer is a nucleic acid. The monomer is a nucleotide. You should know the sugar and 4 bases found in DNA. Compare those to the sugar and 4 bases found in RNA. Given a strand of DNA nucleotides, you should be able to use complementary base pairing of the nitrogenous bases to replicate that DNA. So using each strand of our double-stranded molecule as a template, you should be able to construct the complementary strand that goes with that. So you end up with 2 identical DNA strands, so new DNA molecule. So that's replication, making a copy. That happens in the S phase of the cell cycle. That's how we go from a single unduplicated chromosome to a single duplicated chromosome consisting of 2 sister chromatids. Identify the functions of messenger RNA, ribosomal RNA, and transfer RNA. And then given a template strand of DNA with the nitrogenous bases, you should be able to use complementary base pairing in transcription to construct a messenger RNA molecule. And then from that messenger RNA, using the codon chart, you should be able to translate that into a polypeptide, a string of amino acids. So first, let's look at our nucleic acids. The 2 that we are primarily concerned with are DNA and RNA, deoxyribonucleic acid, ribonucleic acid. These are similar. They're both nucleic acids. They're built of the same monomers, similar monomers, but they do have some differences. DNA is a double helix. It is a polymer made of many nucleotide monomers. Right? So my single Lego is my monomer, and that is a nucleotide. And then I stack all of those together, all my Legos. I can stack into different arrangements. If I stack 2 parallel stacks of those nucleotides of Legos and then twist it, I have a double helix. That's DNA. My RNA is a single strand, so I would just take those repeating bricks, my monomers, and just make a single stack of those. So a single strand. It's also a polymer made of many nucleotide monomers. So they're similar. They are polymers. They're built of repeating units of monomers. Remember, monomer is my single brick. Polymer, lots of those bricks stacked together into a wall or a house or whatever else you might stack your bricks into. If we continue to look at the similarities and differences, the sugar in DNA, giving it the D, deoxyribonucleic acid, the sugar is a 5-carbon sugar. So it's a pentose sugar called deoxyribose. In RNA, ribonucleic acid, that R is for ribose, also a pentose 5-carbon sugar. So pentose means 5 carbons. So they're both 5-carbon sugars. This is deoxyribose. This is ribose. They both have a phosphate attached and a nitrogen-containing base. So we say a nitrogenous base, we mean it contains nitrogen. So that's my building block. That is my single Lego monomer. is this nucleotide made of a 5-carbon sugar, a phosphate, and a nitrogenous base. The difference is my sugar is different, deoxyribose versus ribose. My nitrogen-containing bases, those nitrogenous bases here, can be adenine, guanine, cytosine, or thymine in DNA. In RNA, I don't have thymine. I have cytosine, guanine, adenine, and uracil. So my T is substituted for a U, uracil. Right, and then I stack those together. So each DNA contains deoxyribose, nitrogen-containing base, it's either adenine, thymine, cytosine, or guanine, and a phosphate. RNA, each nucleotide contains a ribose sugar instead of deoxyribose, and the nitrogen-containing bases are adenine, uracil, cytosine, or guanine, and also a phosphate. My DNA, you can think of it as a ladder shape, which has a backbone. The uprights of the ladder are my sugar, my deoxyribose sugar, there it is, and the phosphate. Those go up either side. Here, remember it's anti-parallel, so you see that sugar is oriented. Here I'm oriented with 1, 2, 3 on the bottom, 4, 5th carbon on the top. Here I'm oriented with 1, 2, my 3rd carbon on the top, 4th, 5th carbon on the bottom. So anti-parallel to each other. So that's my backbone. And then the rungs of the ladder, or the steps, would be the nitrogenous bases. And they have complementary base pairing. Adenine always pairs with thymine. Guanine always pairs with cytosine. Those nitrogenous bases pair with hydrogen bonds. So some of the reasons, I always have a purine with a pyrimidine. If you see, my purines, adenine and guanine, are kind of 2 ring units long. My pyrimidines, thymine and cytosine, are 1 unit long. In order for my ladder, for those uprights to be parallel, the 2 nitrogenous bases, when paired, have to be the same distance across. If I paired guanine and an adenine, I would get a bulge because these 2 together would be bigger. So this wouldn't be parallel. If I combined a thymine with a cytosine, this would narrow. It wouldn't be as wide across because those are only single units. So I have to have a purine with a pyrimidine. And the reason I won't get guanine with thymine, this purine with this pyrimidine, is because here I have 3 positions that can form hydrogen bonds. And on thymine, I only have 2. So guanine can form 3 hydrogen bonds. Cytosine can form 3 hydrogen bonds. Thymine can only form 2. Adenine can only form 2. So they pair up, because if I tried to connect my thymine to my guanine, there'd be this spare hydrogen bond. This hydrogen would be looking for some place to bond, and it would give me a kink in my ladder. So that's why my thymine and adenine always pair up, and my cytosine and guanine always pair up here. And they are connected by those hydrogen bonds. What holds my 5-carbon sugar, my backbone, together, the 5-carbon sugar with a phosphate, is covalent bonds between the phosphate of one nucleotide and the 3-carbon of the sugar in the next nucleotide. So those are covalent bonds holding my monomers together in the upright. So from here to here to here to here, covalent bonds. And across, holding my 2 strands together, are my hydrogen bonds. So here we see that hydrogen bondings, 3s and 2s. There's a single nucleotide. And then because of that bonding orientation, this is going to curve. So it's like a ladder that somebody took and twisted. All right, that's my DNA. My RNA, I have 3 different types of RNA that are going to be involved in transcription and translation. RNA is a single-stranded molecule, so my sugar phosphate is still the backbone, but it's a single sugar phosphate with nitrogenous bases on the sides. Right? So we still get that helix shape, but it's only a single strand that kind of coils. Whoops, where'd we go? Here we are. I have cytosine, guanine, adenine, and uracil, no thymine. And I get these by using my DNA as a template with that complementary base pairing. So my RNA polymerase is going to come along, and when it sees a G, it's going to add the complement of C to build the RNA. If it sees that C in the DNA, it's going to add a G in the RNA. If it sees a T in the DNA, it's going to add an A in the RNA. And if it sees an A in my DNA, then it's going to add a U, uracil, when it builds mRNA, because my RNA does not have thymine. So that's my messenger RNA structure. And we're going to look at the different ways RNA functions. We've got 3 types of RNA. We've got messenger RNA, which is transcribed from my template strand of DNA, and this carries the codons. These are the message that tells me what order to put my amino acids in. What amino acids go in what order? My rRNA, ribosomal RNA, is again a single strand like this of RNA coiled around proteins to give me this shape, to give me my ribosome. So it's a structural component of my ribosome. My tRNA, again, also made of the same material, RNA, but this single strand would fold up on itself into this specific shape that gives it a hook to grab onto a specific amino acid, and these 3 positions here that are the anticodon, or the complement to the codon that tells me what amino acid goes where. This works like a lock and key. The anticodon are the complements to the codon, so any tRNA carrying its specific amino acid can only latch on where its anticodon is the complement to the codon. The codon tells me the order in which amino acid goes there. The codon table is used to translate the messenger RNA. All right, so let's look at the whole process. DNA replication. Replicate. If you see DNA replication, we are not talking about building a protein. We're not anywhere close to there. We are making a copy. Replication means make a copy. So we're going to use complementary base pairing, and we're going to make copies. So I have a double-stranded molecule. I enter the S phase of the cell cycle. It's time to make an exact copy, a Xerox copy that's going to be my sister chromatid. How do I do that? Well, I have a double-stranded molecule, and I need 2 of those. So replication, helicase is going to come in, separate out my double strands. DNA polymerase is going to come in and use complementary base pairing to build the complement using that each strand as a template. So I end up with 2 double-stranded molecules. So 2 DNA molecules held together at the centromere. I have duplicated them. I now have identical sister chromatids, the Xerox copies, in that X shape in my nucleus of my cell. DNA replication, that is the S phase. I do that so that during mitosis, I can separate out those copies into 2 daughter cells. So let's practice that. Use complementary base pairing. And I don't really talk about codons in the DNA, but when we get to the messenger RNA, that's how it's going to be read. So it's just kind of easy to divide these up. So go ahead and what would your base pairing be? Thymine always pairs with adenine. Adenine always pairs with thymine. Cytosine always pairs with guanine. Guanine always pairs with cytosine. And so there are our base pairs. Transcription and translation. These are always, replication is always presented with transcription and translation, 2 completely separate things that we're going to do with our DNA. Replication is the S phase of the cell cycle, when I am going to make a copy so that I can divide into 2 daughter cells, and each one will have a complete set of all the information the cell needs. Transcription and translation occur during the G1 and the G2 phase. During G1, the cell has a job to do. It's functioning. That function will require production of things. It's got a job to do. It's doing its job. So we need to make the proteins or the enzymes that are going to allow metabolism to happen, the cell to function. So I'm going to build proteins. In G2, in order to prepare for mitosis, I need to make my proteins and get ready to divide. So proteins that are going to organize my DNA, wrap around those histones. So I need to produce those. So that's what I'm going to use transcription and translation to make proteins. All right. Transcribe, if I transcribe something, I am going to write it down in another form. So the court transcriptionist, this court stenographer, takes verbal words and writes them down in a transcript of the conversation of what went on in court. Transcription in DNA, I'm going to take the information in DNA and I am going to write it down as RNA. So that's transcribing. So when we transcribe, we use DNA. The template strand of DNA will make a complementary messenger RNA molecule. I'm going to use complementary base pairing. So here's my DNA molecule, and this is the template strand of DNA. Since this is the template, it means it's the one that I use RNA polymerase to make my messenger RNA, and I use complementary base pairing to build my messenger RNA in codons in groups of 3 is how I'm going to read it. Again, we still don't need that, just makes it easier to do this if we divide it up. But when I transcribe now, A's get used. So my T always is the template for A, for adenine. My adenine is the template now because I'm building messenger RNA for uracil. My cytosine codes for guanine, guanine for cytosine, guanine, guanine, adenine. So thymine in my DNA still codes for adenine. So I always use the complement from one strand to the next. And when I transcribe this, I get AUG, CGG, AUA, blah, 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 just to show this is going to be a really long strand. And my final codon is UGA. So now I have my codons, my triplets, 3 nitrogenous bases that I read together, and I use the codon table to translate these into a protein. So translation happens at the ribosome. The messenger RNA, ribosome's 2 components latch onto the messenger RNA and the ribosome reads the messenger RNA in triplets, codons. The tRNA carries an amino acid in, drops it off onto this growing polypeptide. As the messenger RNA moves down the strand. It just plugs along reading codons. And my tRNA comes in, and the rRNA is just a component that builds. It's part of this ribosome. So if I translate something, I turn it from one piece of information to another, from one language to another. I am going to translate from my code of nitrogenous bases into amino acids. So I am translating this into this new language. These were my codons, right? So there's AUG, and I can come up in this table and find my first letter over here is A. And then across the top, my second letter is U. So look where those two intersect. And then over here on the row that they intersect in, I find my final letter, AUG. Methionine. Methionine is always the start codon. It can also be in the polypeptide chain, but there's a specific marker that designates this as the start of my polypeptide. I could use the wheel if you want to use the genetic code wheel. You start on the inside, you find your first letter on the innermost circle. So let's do that for this one, C. All right, so now I know I'm in this quadrant between 3 o'clock and 6 o'clock. If we look at this like a clock, there's my first letter, my second letter, and the next next ring outward from that, so a G. So here's my G. So now I know I'm only looking between 5 and 6 o'clock here. And then I find my third letter in the final one, which is another G, arginine. So I continue, methionine, arginine, isoleucine, on and on and on until I get that stop codon. Remember, I have three stop codons, UTG, UAG, and UAA. All right, so your lab. Lab 13 and 14 are both due on the same day. So this is just kind of your review from chapter 3 when we learned about our biological macromolecules, and then builds on that with information on how we make a copy in the S phase, and then how that DNA goes, or how our DNA goes from nitrogenous bases, all this information into actually building a protein. So gene expression, one gene, one protein. So while look over the PowerPoint, watch the video, do your pre-lab, your videos, your top hat information and activities. You can email nope, those were the Punnett squares. You could email me. Complete the activities, do your virtual lab, your DNA biology and technology. You're going to isolate DNA, and then your post-lab activities. Quiz 11 covers both labs 13 and 14. The link to that is in the lab 14 section of D2L. All right, so that's it for this, and I will have lab 14's presentation posted shortly. Thank you all.